Hello, everyone. Thank you for joining us for our third installment of TDT Talks. We really appreciate you taking the time to hear our speakers today. I'm Jessica Falcone from TDT. I'm on the technical sales team, and I'll give a very brief introduction to our speakers and I'll let them take it from there. So today, we have Gavan McNally, who's a professor of psychology and neuroscience in the School of Psychology from University of New South Wales, and with us presenting the talk today is his research associate, Philip Jean-Richard-dit-Bressel, and he is also at the University of New South Wales, and I will let you introduce yourselves.
Excellent. Thank you, Jessica. Greetings, everyone. I hope that you are well in what remains a difficult time. It's a pleasure to welcome you, virtually at least, to UNSW Sydney. Thanks for joining us, especially if your time zone is a difficult one. We fully understand the problem of meridians. Thank you also to Jessica and TDT for inviting us to take part in this series. Today, Philip Jean-Richard-dit-Bressel, a postdoctoral fellow in my lab, is going to take you through some recent work that we've been doing on the Mesolimbic dopamine system, specifically in understanding relapsed alcohol seeking. And the goal is to, in part, describe what we do with our photometry data, as well as why and how we do it. The relapse work, which is going to serve as the vehicle for talking about our approach to photometry, has been led by Sylvia Liu, a PhD student in the lab. Sylvia has completed a PhD and she's very keen to undertake a postdoc in North America. If you're interested and if you're after someone who can do all the different things that you're going to hear about today, Sylvia would be a fantastic addition to your lab. I recommend it to you very highly. I'll hand over to Phil now for his presentation, and then I'll see you at the end to help moderate and maybe answer some questions. Thanks, Phil.
Great, thanks, Gavin. So again, thank you, TDT, for the invitation. Thank you to everyone for tuning in. So I'll be covering some of the photometry that we did in our recent JNeuro paper shown here. And before I get into it, I really do have to acknowledge the people involved, in particular Sylvia. So this paper was essentially her research. She performed or had a hand in absolutely everything that I'm about to present, from doing surgeries to running animals, analyses, and helping to write code. So she can do it all, and I cannot overstate her contribution to the presentation I'm about to do. And yeah, as Gavin said, she's actually just submitted her thesis. She's, if you're interested in hiring a really fantastic postdoc, feel free to contact her or us if you have questions. And I want to thank other members of the lab that were involved: Ash, Joe, John provided electrophysiology for the paper, and Con Clifford has been a long-time collaborator in developing our photometry analyses. So for some backdrop, one of the interests in the lab, it regards the neurocircuitry of addiction and relapse, and we study it using a rodent model. So rats are initially given an opportunity to self-administer beer in acquisition phase in context A, and across this phase they acquire high rates of nose poking. So that's the seeking response for beer, to the point of intoxication on many sessions. And then we extinguish this behavior in context B in extinction phase. So when the beer is no longer available, in our version of rat rehab, responding decreases. And then when we test them in context B, what we see is responding is low, so extinction has been effective. However, when we test them in context A, their original acquisition or self-administration context, responding returns. And this models the role of context in triggering relapse behavior. And to model the scenario where seeking does yield the drug, as is, is probably true in real world contexts, we basically give them a reacquisition test. So these sessions are identical to self-administration, and when the beer is available, we see a profound return of responding akin to full-blown relapse. So it's well established that dopamine is involved in relapse. In this paper, what Sylvia found was if you inhibited VTA dopamine neurons, either chemogenetically or using optogenetics, you can attenuate relapse. And likewise, if you systemically administer dopamine 1 antagonists, or if you even just administer dopamine 1 antagonists into the accumbens, you attenuate relapse as well, both renewal and reacquisition. So it seems that, it seems that dopamine neuron activity and dopamine release are driving relapse behavior. So our question, or one of the questions we had, was what are the patterns of activity and dopamine release across the mesolimbic system that might be mediating this drug-seeking behavior? And to get at this, we used fiber photometry. We looked at VTA dopamine neuron activity using GCamp in TH-Cree rats, and we also used D-light fiber photometry to look at dopamine release in different parts of the accumbens. And specifically, we wanted to understand what are the activity patterns associated with seeking and consumption. And one way that we got at that question is using confidence intervals on our event-related activity. And so I'll cover that in a few slides. Another question that we had is, how do these activity patterns change across phases of seeking? And to get at that question, we used conventional area under the curve based analysis, but we also used a analysis strategy called representational similarity analysis, which I'll be going over towards the end of this talk. So kind of getting into the nuts and bolts of it, our photometry rig kind of follows the same conventional framework laid out in Lerner and colleagues. So we have an isobestic control signal that is used to correct for motion artifacts. And our real-time modulation and demodulation is taken care of by TDT's RZ5P and Synapse software. And our event timestamps that are occurring within our mid-associates chambers, all of that is being transmitted to the TDT RZ5P. And so basically everything that we need to analyze our data is stored in those Synapse storage tanks. The rest of our gear, all the LEDs and the photodetectors are from Doric, and we actually make our own patch cables and implants. So all of this, the photometry rig is mounted onto a mobile server rack, so we can wheel our photometry setup into the rooms that's needed, including our self-administration chambers for our addiction-related research. So after we've run our photometry experiment, this is essentially what we have stored in Synapse, right? We've got our signals, we've got event timestamps, but we need to do more with that. We need to analyze it. And to do that, I developed a kind of a pipeline of analysis, and this comes in the form of a suite of MATLAB scripts. This is kind of a page from the manual. And all those scripts do is essentially import the data into a standardized structure within MATLAB. We input any additional details needed, like subject or session IDs, or even if there are disconnections during the session, we can log those times as well and basically say we're not going to analyze those portions of the session. We do some signal processing, which I'll cover on the next slide. And then finally, we can get around to actually analyzing our signals for event-related activity or doing more complex things with it. So as I mentioned, we do some signal processing, and the general idea around signal processing is that we want to remove any problematic signal components or any unwanted noise or any unwanted trends in the data. So for this particular experiment, we are looking at phasic activity around events on the order of seconds. So to remove longer-term trends in the signal that might be, which are essentially not needed for our analysis, we use high-pass filtering. So we basically get a moving average of our signal, and we just subtract that moving average, and that straightens out the signal, removing any longer-term trends in the data. To evaluate any high-frequency noise in the signal, we use fast Fourier transform, and we can detect whether there are spikes. So let's say here there's this spike just above 2 Hertz, and we can remove that using a low-pass filter, or if we wanted a band stop filter. So once we've processed our signal, we're ready to look at event-related activity, and we do that like everyone else. We collate the delta F on F around our events of interest. And of course, at this point, you're kind of faced with questions about how do we normalize our signal? Do we baseline it to some pre-event period, or do we Z-score it? One way that we dealt with, or one way that we kind of normalized our data was using kernels. So kernels are essentially a normalized waveform, and it's like a Z-score except relative to 0 rather than its own mean. And this kernels essentially help us tap into the shape of the waveform and cut through any differences in magnitude, whether they're due to signal differences or changes across a session. So if there's a particular pattern of activity around an event, we can get at it using kernels. So to analyze that activity, a common method is to obtain some kind of simple statistic that's more amenable to traditional analysis, like an area under the curve or a peak delta F on F. And that's fine, and we do make use of AUCs. But the problem with that is that you do lose potentially important temporal information, right? So you've got this waveform, which is a relatively rich piece of data, and the AUC simplifies that, but it also overlooks some more interesting things that might be happening in the waveform. And another thing that's another problem with AUCs is that the analysis window, so the window that you're choosing to derive an AUC from, is usually chosen post hoc. And apart from adding an inconvenient step to the analysis, because you need to pull out a figure and evaluate where you want to take your AUC from, it is a little bit dubious, from an analysis perspective, that you're choosing what to analyze every single time. It opens it up to data mining. So one way that we've kind of gotten around those issues is to instead analyze the entire peri-event period using a waveform confidence interval. So basically, we get a 95% confidence region for peri-event activity. So we can infer when in the entire peri-event window activity is significantly different from the null. And I've shown, using Monte Carlo simulations, that you can control the type 1 error rate by stipulating a minimum duration of significance before you call whether there's a transient present in your window or not. So we get our confidence interval by bootstrapping it. And so bootstrap analysis is statistically powerful, and it doesn't rely on any parametric assumptions, which is nice. But it is, it's worth mentioning that it is a computationally demanding way of analyzing or developing a confidence interval. But most modern computers, can handle that no problem. So the way that you bootstrap, or bootstrap, basically involves randomly resampling from your data set with replacement and re-deriving the estimate repeatedly. So for example, if you have 10 waveforms, you randomly sample from those 10 waveforms 10 times, you calculate a mean, and that is your new bootstrap estimate. And you do this a lot of times, let's say 1,000 times, to derive a distribution of those bootstrap estimates. So if there's a lot of variability in your data set, there's a good chance that you're going to pick from your worst waveform, let's say 10 times, and that's probably going to affect it's going to indicate that there is nothing significant happening in your data. Or in the case where something very robust is happening, you are probably going to detect a transient because your data set indicates as such. So you can actually get the confidence interval from the percentiles of your bootstrap distribution. So it's worth mentioning that the percentile confidence interval needs to be adjusted slightly by a factor related to n, and we take the easiest option. We just expand our interval by this factor, and as I mentioned on the previous slide, you can also control the type 1 error rate by stipulating a consecutive threshold. For us, we use a window equal to the low pass frequency, and we've shown, in Monte Carlo simulations that actually does a good job of controlling the error rate, and it's actually very effective at detecting transients that are present within your window. So all of that is in the Frontiers paper that we published earlier this year, and the code that we use for bootstrapping, I've put that up on GitHub, so feel free to check that out, and I plan to continue adding stuff to that repository as I get to annotating code that might be useful to other people. So going back to the actual data and the actual experiments that we conducted, using that approach, we measured VTA dopamine activity across phases of alcohol seeking, and here we show that activity around the seeking nose poke response, specifically the nose poke response that turned on the pump, yielded significant transients in the VTA. And interestingly, we are finding different patterns of activity in the medial versus lateral VTA, which fits with recently emphasized dichotomy between these two portions of the VTA. Specifically, we're getting positive transients just after the nose poke across all sessions for the lateral VTA, whereas for the medial VTA, we see a flip from an inhibitory transient to an excitatory transient across acquisition. We're not getting any significant transients during extinction or ABA, but we do see excitatory transients in reacquisition. We also looked at dopamine release within the accumbens subregions using D-Light, and I think in the interest of time, I won't go into all the little findings, but basically we're getting different profiles of activity across the phases depending on the subregion that we're measuring from. And we'll have time to talk about that later. So, apart from characterizing which mesolimbic compartments are showing event-related activity transients, during which phase and during what, at what time points around that event, the broad takeaway, from all of that was that, different things are happening depending on the phase as well as the subcompartment. But, one question is, how does activity change within a region across phases? And, if you used AUCs, well, there are 10 pairwise comparisons across our 5 stages. And, and that's only using one AUC window, which might not give a full enough picture of what is actually going on. So we are immediately, if we're using conventional event analysis, we're immediately running up against, I guess we're running into some problems. Additionally, if we wanted to make comparisons between different regions and sensors, we run into some, rather thorny validity issues because there are, likely differences in expression levels, and there are differences in sensor dynamics that make AUC comparisons unfair. So given that issue, we were trying to think of ways to actually get around those problems and actually make comparisons between regions, sensors, and across phases in a succinct way. And fortunately, this problem has been solved. So Colin Clifford pointed us to an existing solution called representational similarity analysis. And a notable person who's developed this technique is Nicolaus Kriegers-Rother. So RSA is essentially a method to summarize and compare the similarity of neural activity across multiple domains. And those domains could be phases, could be brain regions, could be measurement techniques. So the first thing that you do is you get the activity that you can reasonably compare. So in our case, these are the mean kernels of activity in a region across sessions, and we can compute a similarity score between these waveforms. So we used a Pearson correlation between the waveforms to compute their similarity. So let's say early acquisition and late acquisition nosebud waveforms are really similar from a correlation standpoint, whereas the correlation between late acquisition and extinction skews positive but is less strong. We then compute or convert this score into a correlation distance by subtracting it from 1. So a score of 0 means that the waveforms are identical, whereas a score of 2 means that they're perfectly anti-correlated. And we can plot those interrelationships in a matrix or an RDM that summarizes how similar nosebud waveforms are across the experiment. So one way to kind of visualize these relationships is using multidimensional scaling, or MDS. And basically what that does is it attempts to represent all those distances in the matrix within some lower dimensional space, in this case, a 2D space. So here we're plotting how dissimilar sessions are from each other. And it kind of intuitively conveys how all your acquisition sessions and your reacquisition sessions are very similar to each other, relative to renewal and extinction. So when we do the same thing for MVTA activity, it highlights how dissimilar early acquisition is from late acquisition, so in stark contrast to the LVTA, as well as the rest of the how different early acquisition is from the rest of the experiment. So these are the RDMs and MDS graphs for the incumbent subregions. And again, I don't think, well, you can check out the paper because we discussed this, in some detail there. But I guess the broad takeaway is we're getting very different looking RDMs across our subregions. And what that tells us is that these regions are not treating these phases the same way. So the way that, so those first order RSA or first order RDMs are a nice way to actually summarize how activity, like what activity is similar across the sessions, but it doesn't actually allow us to directly compare between regions. And this is where second order RSAs come in. So a second order RDM is actually a dissimilarity matrix of the dissimilarity matrices. And the logic behind second order RSA is that first order RDMs are an abstraction of the underlying data. So, matrices from otherwise incomparable data types, can be compared on a level playing field because they're all converted into this RDM. The logic is exactly the same. We can compute the correlations between our matrices, and we can basically create another RDM, except this one being a second order matrix. Now, it's worth emphasizing that we are not directly comparing the underlying data itself. We are comparing RDMs in this second order matrix. So, imagine we had two regions with perfectly opposite activity patterns, right? So this region 2 is just the flip side of region 1 data. So they have perfectly opposite activity patterns, but if we were to calculate a first order RDM for region 1 and a first order RDM for region 2, what we would get is the exact same dissimilarity matrix, because the interrelationships or the similarity across phases would be exactly the same. And so when you input those two first order RDMs into that second order RDM, that second order RDM is going to say that these two regions are representationally identical. That is, they're kind of encoding or representing the task space in exactly the same way, right? They're treating these two sessions as the same, and this session is different from those first two. So in terms of the, interpreting this actual dissimilarity matrix, across our mesolimbic compartments, we're basically observing similarity between the accumbens shell, the medial accumbens shell dopamine release, as well as the neural activity in the MVTA, as well as LVTA neuron activity and dopamine release in the accumbens core, which fits with those projections. So, it's essentially showing the functional connectivity between these two regions, or these two pathways. So because we actually also did a second order RSA for each phase of the experiment, and we can kind of do this because the parameters for recording across our regions were actually congruent. So, we had the same sampling rate for our VTA recordings and our D-Light accumbens recordings, and so we could compute a correlation score of, VTA responses to the nose poke versus accumbens responses to the nose poke. And as I mentioned before, there are concerns with reading into those first order comparisons because there are issues of correspondence between, let's say, D-Light and GCAM6F, though it is worth saying if there were two different sensors that you wanted to compare with each other, GCAM6F and D-Light aren't the worst two things to compare. But that being said, we can still overcome that issue of correspondence by looking at or computing a second order RDM based on those first order comparisons, because those first order RDMs across your phases are now on a level playing field. They're making the same comparisons across regions. And I guess some interesting takeaways is that early acquisition is representationally dissimilar from late acquisition across the mesolimbic system. So basically it's saying there are differences in the differences between these two phases. And also quite interestingly, reacquisition is relatively similar to late acquisition. Despite extinction. So after late acquisition, animals are extinguished, and instead of returning to a profile of activity that resembles early acquisition, animals, when re-exposed to the drug, immediately return back to signatures of established self-administration. So that kind of fits with our understanding of the ingrained nature of drug seeking, that it is resistant or it's not, it's not nullified by extinction or loss of responding. And I guess pertinent to our questions about the neurocircuitry of relapse, we see distinct signatures for our two different forms of relapse, renewal and reacquisition, which suggests different roles for mesolimbic compartments in each of these different forms of relapse. And that actually fits quite nicely with, that's corroborated by the manipulation data the distinction between medial versus lateral VTA that I actually showed at the very beginning of this talk. But check out the paper if you're interested in looking into that. So wrapping this up, in quick summary, we measured dopamine neuron activity and dopamine release across mesolimbic compartments, across phases of alcohol seeking, and using a multivariate approach called representational similarity analysis, we observe pathway-dependent and extinction-resistant profiles of activity. So thank you all for listening. Once again, I must acknowledge Sylvia and other members of the lab, collaborators, and funding bodies for making this work possible. And thank you all for tuning in.
Yeah, excellent.
Thank you very much. It's very good. All right, just in case anyone has any questions, you can either enter them into the chat, or there is a questions tab where you can type that in, and the floor is open for Q&A right now, so please ask away. And then just as a quick follow-up to, you know, if there's more things that we're not quite able to get to, we can do a follow-up coffee hour next week. We'll pick a time and send an email out, so just keep a lookout for that. All right.
So Phil, there's one question there, and you almost hinted, well, you hinted at this when you talked about the difficulties in the RSAs on D-Light versus GCamp, and that is the low-pass filter. Did we use the same low-pass filter for D-Light and GCamp? And I guess embedded in that then is, if we didn't, why didn't we?
I believe that a different low-pass filter was used for GCamp and D-Light experiments. So
3 Hertz versus 2 Hertz?
Yeah, so the GCamp data was low-pass filtered at 2 Hertz, whereas the D-Light data was low-pass filtered at 3 Hertz. And I believe the reason for that was, well, on a conceptual level, D-Light has a faster kinetic profile. So you might be filtering out some of what, you know, signals, actual dopamine release signals, if you low-pass filter too low, whereas there's less, I mean, granted, GCamp also operates within the same, you know, on similar timescales, but the signals that we're getting out of the VTA were massive. They were over the course of seconds, whereas the D-Light ones tend to be much quicker. So I believe there was a decision that the low-pass filtering would happen at 3 Hertz. And again, that contributes to the correspondence between those two sensors, whether we can make direct comparisons between what's going on in those D-Light experiments versus those VTA experiments without RSA or the second order RSA.
Yeah, good answer. Great question. We have another question. Oh, hello, G. Have we tried examining early versus late extinction to understand the retrieval of memory differences to extinction? I can answer that one. Well, we have. The difficulty here in doing that around the photometry, at least, is, as you well know, is that the behavior dies, right? It goes away very quickly. So by the time you get to the end of extinction, if your extinction's been any effective, and it almost always is effective, you don't have enough events at the end of extinction to be able to look at transients. They're just, the nose pokes go down too much. So we have tried, we just don't see enough behavior. You could ask, I guess, it's difficult because I'm just reading a question. I guess you could also look within a session. You could look at how much the start of the session versus the end of the session. And we have looked at that, but again, the difficulty is that behavior is always front-ended in this task. So the behaviors in extinction is almost loaded at the front of the session rather than at the end. So you're running up against this wall of how many events do you actually have at the end of either extinction training altogether or at the end of an extinction session in order to make a comparison between the two phases. I hope that answers your question, G. Oh, we have Lizzie from Newcastle. Hello, Lizzie. Phil, can you quantify differences in the RSA analysis? Can you do stats on that? Bootstrap, for example?
Yeah. So that's a really good question. And on one hand, we have the, it's quantified by the actual correlation distance. So on one hand, we have a fixed point estimate. Whether we can get a confidence level on, or a confidence interval on each of those correlation distances. Yes, theoretically you can. You could, let's say, do some kind of shuffle or some kind of bootstrapping to assess where those differences are and say, okay, are the similarity or dissimilarity between this region and that region, or this phase and that phase is somewhere in this region. But again, that's bootstrapping, and if you imagine trying to bootstrap all of those waveforms 1,000 times and take it to the second order RSA, that it's a tall task, but it is definitely possible, and it would be cool to develop, incorporate that into our pipeline.
Yeah, the Krieger's Court papers in PLOS biology and computational biology outline a number of different ways, Lizzie, that you could make comparisons on the second order RSAs. That approach to comparing disparate data types or data from different sensors or data from different recording methods is used quite extensively in cognitive neuroscience. So there are a number of different ways in which you could do that, and if you're interested, I would encourage you to have a look at those papers, because they're really very good, and they're quite readable. We have a question from Max. Apologies if this is in the paper. Do you have and/or compare a control group's waveforms using NIRBE? Good question. If so, is there a difference in waveforms? I can answer that question. We don't. I'd be interested to know if there was in fact a difference in the waveforms. We ought to do that. We get that question a little bit. We used to use NIRBE controls all the time, and there are important differences between the NIRBE and between the BEER. A lot of what we find in terms of both the circuits and in terms of the pharmacology overlaps extensively with what other people have reported using intravenous cocaine, using intravenous heroin as well. So it would be really nice to do that. It would be equally, I think, important to see how it is that either NIRBE or alcoholic beer differs or does not differ from calorie-matched sucrose, and also how it does or does not differ from intravenous cocaine. I would be especially interested in that for the medial accumbens shell, because in the medial accumbens shell in the paper, what we consistently find is a decrease, a dip or negative transient in the D-light, in the dopamine binding. And it would be very interesting to see if that's simply related to consumption and therefore independent of oral ingestion of what's being ingested. Do you see it with sucrose? Do you see it with anything else? Or do you see it in something without oral ingestion, like intravenous self-administration? So I don't know the answer to your question, Max. It's a very good question, though. Thank you.
Yes. Are there any other questions? Because that is all that we have right now. So speak or forever hold your peace. No, well, I was going to ask something in the similar vein as the last person, so I'm all out. All right. Well, I guess if that's it, we can go and wrap up a little bit early then. So I really want to thank Phil and Gavin for taking the time to speak with us tonight. Oh, there's a couple more coming in, but those are more. Yeah, everyone's saying thanks for the great presentation. Yeah, so I want to thank you guys for taking the time to speak with us. So it's my tonight, your next morning, and thank everyone for logging on. So thank you very much, and have a good day.
Thanks, everyone.
Thank you. The answer is yes.